hex is 6, right? So it'd be like this. That's hexane, 6-carbon hydrocarbon. So take an animal, you blend them up, and I didn't know we got one of these things. I'll show you really quick. I'm using this as a profit class today. This is the classic Waring blender. It's like a commercial blender. We got it for, we're not grinding up animals. Just take a mouse. Let's say you want to know the lipids in a mouse. You put a mouse in the blender. You blend him all up.
Oh my gosh.
Okay, a hot dog. Hot dog? Take a hot dog. Gee, where'd the hot dog come from? Did you see that? The recent news, there's been two recent articles on hot dogs. One was that when they label a hot dog vegetarian or they label it all beef or all chicken, there's usually like some other thing in it. And so most of the hot dogs have something other in it. One of the other things they have in them, human DNA.
Yep.
Yeah, somebody gave you the finger, literally. Chopped it off, threw it in there. Disgusting. Anyways, it's probably not from that, I'm just saying.
Where's it from?
Huh?
Probably from hair.
Hair doesn't have DNA, does it?
Yeah.
Hair roots do. The hair itself. But it was like 5%.
Probably like a fingernail.
5%. So I was trying to think, what's 5% of my body? That's where I came up with the finger. Lost the finger. That makes sense. Anyways, they would put tissues in here, blend them up, put it in hexane, shake it, and everything that dissolves, that's a lipid. So you can imagine if you've had biology, all the nonpolar things that would be in there. And that's what this chapter is about. It's about everything that dissolves in hexane. Okay, so lipids. So a couple of things that we have to be able to do from chapter 12, actually, is hydrogenation reactions. But we know that. I'm going to pass over hydrogenation, but that's the adding of hydrogen, H2. Okay. You know how to name alcohols. Naming carboxylic acids, recognizing esters, and hydrolysis of esters. These are all things that if you need to go back and review, those are the sections in the book to go back and review. I won't spend too much time on it because we can mostly work around that stuff. Okay, so lipids are biomolecules. Well, that's the important phrase there. Hang on. I'm going to put my highlighter on. Biomolecules that contain fatty acids. So fatty acids are carboxylic acids or a steroid nucleus. So this thing on the right is the steroid nucleus that you're looking at, okay? They're soluble in organic solvents, and like I said, originally it was whatever you could dissolve into hexane. So you can imagine that the steroid molecule, which is the nucleus of cholesterol and all the hormones, the sex hormones, okay? That this thing would be soluble in a nonpolar solvent because there's no oxygen in here at all. It's just all carbons. It's a hydrocarbon, okay? And the Greek word for lipos means fat, okay? So fat is lipos. These are, you find, because of the nature of how you find them, okay, they're in cell membranes, they're molecules in your in intracellular and extracellular fluids, and they're also hormones. I think the gist of this is that it's all kinds of stuff. You don't have to remember particularly any kind, but you should remember it's in cell walls, right? There's some vitamins, and there's steroids. So let's go ahead and talk about the different kinds of lipids. And again, this is a hodgepodge of things, and we'll try to work our way through these things. Probably the most important type of lipids are the fatty acid lipids, okay? So what is a fatty acid? A fatty acid looks like this guy down here. That yellow looks really good on there, actually. Sorry, I replaced the book's figure because I couldn't tell what they were doing. And if you count this, we're just going to do this for fun. Well, it's kind of for fun. Where's carbon number 1? It's here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Okay. So this is a typical fatty acid. This is the fatty part. It's just really the nonpolar portion. Not soluble in water, soluble in organic solvents. Okay? And what we're going to do is we're going to talk about each one of these kinds of lipids, triacyl glycerols, glycerol phospholipids probably follows better after this, waxes, and sphingolipids. Sphingolipids you'll find in things like nerve tissue, the myelin sheath, those kinds of things. Those are made out of sphingolipids. So you can imagine, like, when you're talking, like, again, things in the body, it's stuff from everywhere, right? Okay, now, one of the things that these lipids have, these types of lipids have in common is what's known as this fatty acid chain, which is connected by an acyl group. The acyl group is this whole thing that's in yellow, okay? It's including the C double bond O. So when I say it's a triacyl glycerol, right, how many acyls does it have? 3, okay? What if I say it's a diacyl? They should have 2, okay? And in fact, those are the 2 real common ones. You'll see diacyl glycerides and you see triacyl glycerides. If you get your blood work done, they're usually listed as DAGs and TAGs. Okay? So di and tri. We'll go over what each of these things means. They're all esters, okay? This group, in order to make an ester, what do I need to do? What do I replace the H with? A carbon, right? This H gets replaced with the carbon, and that becomes an ester group. So this is going to be these will be basic features of all of these lipids. Okay, so just general. That color doesn't show up at all. Okay, this is supposed to be blue.
It's very light.
Blue.
Very light.
Yeah, sorry. Okay, I won't do that again. Book slides, that's what you get. So glycerol is this. Is it warm in here? It's just me, huh? This is glycerol. Okay? So if you imagine that that's glycerol, then what you have is CH2O, CHO, and CH2O here. And then you have the long fatty acid chains on it. This is what is known as a triglycerol, or a triacyl glyceride. Okay? A diacyl glycerol looks like this, and you'll notice there's 2 fatty acid chains on it. And then there's this phosphate. So I'll show you what phosphate looks like, but there's a phosphate that connects it to an amino alcohol. What is an amino alcohol? What does it sound like? It has a nitrogen and an OH. Okay, so it's usually connected through an OH, so that you have something like this, and then let's say CH2, CH2, NH. And normally we would write NH2, but at physiological pH, and that's what we're going to deal with a lot of, this will be NH3, sorry, not 4, plus. Okay. And that comes from something called ethanolamine. I'll explain that to you in a little bit. So those make glycerophospholipids. This class of compounds includes things like lecithins and cephalins. So I'll explain to you what lecithins and cephalins are. But that's one kind of fatty acid molecule. This molecule consists primarily of like cell wall components and things like that. Fat. All right. Sphingolipids kind of look like this. Instead of having glycerol, and I'll actually show you the structures of these things, but just to get the general scheme, there's a molecule called sphingosine, and
Backbone, I don't understand these drawings. Like the highlighting and everything.
Okay,
Is that
yeah.
what's coming off of it?
Yeah.
Glycerol?
so here, right?
I don't get it, yeah.
Yeah. Let me draw it for you then.
Okay, yeah.
So, but there's a CH2O and then the fatty acid. Another CHO and a fatty acid, CH2O and a fatty acid. So let me draw just like a typical triacylglyceride, okay? So, and it takes a lot of space. Okay, so stearic acid is one of the common fatty acids. Its formula looks like this: CH3.
Tons of like carbons and hydrogen.
Yeah. So let me draw it. Stearic acid. It's CH2. So I'm going to draw the condensed structure for it. There's 16 in between. It's completely saturated. There's no double bonds. Okay. If there's a double bond, I have to draw that. And then it's C, and then double bond O, and then OH. So this is stearic acid, or we'll call it stearate in a second. Oops. So let me ask you a quick question. How many carbons are in stearic acid? 18. So it's a C18, okay? So a lot of times when you see stearic acid, you'll see this little note after it, 18, and then a colon and a 0. This is the number of double bonds.
You asked what? The number of carbons?
Yeah.
Oh.
So a lot of times they'll say stearic acid, and you'll see 18-0. 18-0 means 18 carbons, no double bonds. This is a saturated fat or fatty acid. Saturated fatty acid literally means it can't take any more hydrogens. If it has a double bond on it, it can pick up more hydrogens. So this is a saturated fatty acid. Okay, so now what I'm going to do is I'm going to make the triglyceride from that. That means I need 3 of these attached to glycerin, okay? So you're going to write this, you're going to take space. And I could copy and paste on here, but I'll save you the agony. I'll go ahead and write it. So I'm going to go so I need a glycerin backbone. So I'm going to go like this. And I'm going to do this crazy thing. I'm going to make it really big. And that's only so I can space things out properly. I'll show you there's other ways to do it, but this is the way I think people are most comfortable seeing it. Now I need to get 3 stearic acids on there. And they need to be acyl groups. But they're esters. So that means that's 1. Do you see the stearic acid? Yeah, so, and then I'll have 1 more. It'll be C double bond O, CH2, 16 CH3. I swear I'm writing, but it's really not responding well. And then there's another one, C double bond O, CH2, 16, and then CH3, like that. So this has got a couple of names. The common name for it is tristerin. Because it has three steric groups, three sterates on it, okay? The systematic name is to call it Glycerol. Oops, that's horrible. And then trister. I'm writing, but it's not following along. 8. Go ahead.
I forgot, but what did you say that the acyl group was again?
Yeah, so to say it's a triacyl, so this is a triacyl glyceride, means it has, that's the acyl group.
Oh, okay.
So it's 3 of those, and this part from here, this whole thing here, that's the glycerin. Right, that comes from glycerin. So this is why we call it a triacyl glyceride. It's got 3 acyl groups on it. The composition, like these acyl groups, don't all have to be the same. They could be different. Okay, you don't need to know like how they're different. Okay, you just need to know they can be different. Okay, so it has 3 acyl groups on it, and it's attached to a glycerin backbone. Okay, so triacyl glycerides and diacyl glycerides have glycerin as their backbone. Sphingosine is a much more, I guess, in a lot of ways, complicated molecule, but that's the backbone for sphingosines. Okay. So sphingolipids. Okay, so that's what this sketch means. Okay, this is the glycerol, and then you have the acyl groups from the fatty acids attached. The one I drew was saturated because all of the bonds there are no double bonds, right? It's all single bond. And glycerophospholipids, you actually have a phosphate group in between, and I'll show you how that works in a second. But they have 2 fatty acid chains. Again, they could either be saturated or unsaturated. The number of carbons varies from about 12 to 22. Okay? And it's almost always, in fact, I think it's always even numbers and naturally occurring ones. So 12, 14, 16, 18, 20, 22. Those are the naturally occurring ones. Okay? Okay. Yeah. Now, okay, so back up a second. So glycerols, triacyl or diacyl glycerides, they have glycerin as their backbone. There's another molecule, it's called sphingosine, but it also makes lipids, okay? And sphingosine has this structure that goes up like this, and I'll show it to you in a second, just to get the idea. One of the chains never changes. It's part of sphingosine, okay? And then you have one fatty acid, which you can vary, and then you have an amino alcohol, which you can vary. And the amino alcohol is literally something like this is ethanolamine. Okay? Yeah, sorry, we'll keep going. So steroids, I don't know why it says lipids containing sphingosine at the top. That shouldn't say that up there. Steroids contain this 4 fused ring system. And if you look at the number of carbons, it's relatively easy to remember. This is 6, and this is 6, and this is 6, and this is 5. Okay, so if you have to know that pattern, I'm not going to require that you draw testosterone or draw estrogen or anything like that, but you need to be able to recognize what a cholesterol. This is the nucleus, if you want to think about it. Like an atom has a nucleus. This is the center of every steroid hormone. Okay. Okay. Let's see what else is there. Yeah, there are on the steroids, there aren't any fatty acid chains. Okay, and you remember when we talked about fatty acids before and esters, you can always hydrolyze esters, so these are non-hydrolyzable. They're just what you get out of the molecule or the animal. You can think about the animal in the blender analogy. Hang on. Oh, I think I have more details. Okay. So just an overview, and then we're going to cover a lot of these in more detail. Overview is that we have lipids, and there's fatty acid lipids, and there's steroids, and steroids are in their own whole different class. And then fatty acids give us waxes, triacylglycerols, glycerol phospholipids or diacylglycerols, and sphingolipids. So we're going to go, we have like a section of slides on each one of those, and we'll go over and talk about their characteristics. Okay, so lipids are characterized by presence of fatty acids or steroid nucleus. Which type is described by the following? Contains a fused four-member ring. What's that? Steroids, right? Can you write on these things? Oh my gosh. Well, I didn't even catch the middle letters there. What contains long carbon chains? Fatty acids. Includes carboxyl groups. That's also fatty acids. All right, good. Which of the following is not a characteristic of lipids? May contain a carboxylic acid? No, it does, right? That's okay. Contains a four-ring structure. Those are, steroids, right? And those are all lipids, okay. Soluble in water? No. Soluble in nonpolar? Yeah, those are all. So A, B, and C are characteristics of lipids, and D is not. Okay. So again, we're talking broad classes of compounds and their characteristics.
Wait, are you sure it's not C?
Well, what did I say?
You said it's D, but it's C, isn't it?
Oh, A, B, and D are, and C isn't. That's what I meant to say. I'm sure I said it wrong, since you all looked at me that way. And I'm not married to you. No, that's not the lecture.
Okay.
Yeah.
So steroids are lipids?
Yeah, steroids are lipids. Okay, so let's talk about fatty acids. Actually, I'm going to get to this later. But you can think of it this way. Saturated fatty acids, think of them like the Pringles potato chips, right? They stack up really nicely and make those nice little stacks. Fatty acids that are saturated behave that way. They'll stack together really nicely and form solids, okay? It turns out unsaturated fatty acids don't, like these are like Lay's potato chips, which are actually, if you're going to eat potato chips, I think are better for you, but yeah. Just because they actually have more potassium, they have more of the minerals in them still. I don't think they peel them before they make the potato chip. Yeah. Yeah, so if you're having trouble with, have I mentioned I eat a lot of potatoes?
Yeah.
It's that or rice, right? So I actually had 2 potatoes for my food today and some chicken left over from last week. But anyways, yeah. Yeah, because if I don't do that, I don't get enough potassium.
But it's the skin.
I'm pretty sure it's the
So if
skin.
you peel it, then
Yeah, I think if you peel it, you're getting rid of most of where the potassium is.
You want to take like a vitamin? You can't take like a potassium.
You-
it's
you-
better to eat potatoes. Like,
Eat a darn potato. You can, and like I take a blood pressure pill, and it I lose minerals that way. So
Oh, yeah.
it is helpful to have more, and so I could take a pill.
But I think your body absorbs it better when it's
I'd rather eat a potato. It's a little more gratifying than I got to swallow this gigantic pill that tastes like crap. All right, so draw the condensed or line. So some of the things we're going to learn is we're going to learn how to draw these structures. We'll get to that in a second.
Is that why they tell you to avoid saturated fats?
Yeah, so they tell you to avoid saturated fats because they tend to stack like this, and then they form deposits that way. They've got a very high melting point, so at the temperature you eat them at, they're basically going to be semi-solids, but they can turn into solids as well. So we'll talk all about that. Stuck in your veins, and your blood pressure goes up, and all that good stuff. So fatty acids are unbranched, so they're all linear chains. Get my highlighter out. This is the important part. Unbranched carboxylic acids, typically, but not always, like I said, it goes up to 22, I think. Right? One of the really important ones that we'll look at later is arachidonic acid, and it has more than 18. They're insoluble in water. The book makes a big point of that, but it turns out the salts. Remember we talked about if you hydrolyze and you make a sodium salt or a potassium salt, those tend to be more soluble in water, and it turns out fatty acids which have been hydrolyzed with either sodium hydroxide or potassium hydroxide are much more soluble. And those are actually what we call soaps. Okay? And then saturated has only single bonds, and unsaturated contains double bonds, and it can have more than one double bond. So we have monounsaturated fatty acids, and then what else do we have? Polyunsaturated. And poly can be 2, 4, 5, 6, 7. Some of the ones that we get from fish have like tons of double bonds in them. And in general, the more double bonds it has in it, the better it is for you, okay, nutritionally. Although I think the actual answer is eat less fat. But if you're going to eat fat, eat unsaturated
But
fats.
what about saturated coconut oil? Isn't
Yeah, so we'll talk about that. I'll get there. I don't know. I would be giving you some speculation. Yeah, I'll give you some speculation on that, but I don't really. I'm not a nutritional scientist. I should have. I've got cousins that. I have one who's got her master's degree. She's a dietitian. She works in Kaiser. Her mom was also a dietitian. But I don't ask them those questions. I mean, it's not like I go to family gatherings and say, hey, tell me all about. And then they don't ask me chemistry questions. I don't have to explain organic chemistry. I was at church this weekend and somebody says, oh, I've been studying that organic chemistry stuff. I'm like, I'm sorry. Not my fault you're studying it.
You're like, it's my day of rest.
Yeah, leave me alone. Leave me alone. No, I talk about it all the time anyways. I'm married to a chemist. It's hard to avoid. Okay, so lauric acid. Actually, lauric acid it can be isolated from laurel trees, and that's like bay leaf. Okay, so in bay, you'll find lauric acid. Because guys, anybody here had botany? Bay laurel? Yeah, laurel. Anyways, lauric acid can be found in laurel trees. How many carbons does it have? 12, right? You could count the little balls and sticks up there, or you could just go down here. There's 10 here, and there's 2 more. That's 12. It is a saturated fatty acid, right? This is a condensed structure, and this is the line angle or bond line structure. And you just have to remember every end and every bend is a carbon. So if you're going to do this one, just so you can see that there's 12 here. I actually didn't count it before. I just assumed they did 12, so let's try it. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So that's lauric acid. Typically, we will write them as condensed structures just because you don't want to have to count like this all the time. And it's easier to see what it is when it's written like this, okay? All right. So here are some of the different saturated fatty acids. It's just a long list. I don't think you have to know any of these, other than I would say maybe stearic acid, because that's one of the real common ones. That's why I showed it first. Okay. And you'll notice the notation that they have on here. Right? It's 18 and 0. 18 means what? 18 carbons. 0 means no double bonds, right? So all of these are either 12-0, 14-0, 16-0, and 18-0. Myristic acid, I think what is it? Is part of the name of like the nutmeg plant. But that's one of the sources of it. They take nutmeg, the nut, and actually cold press it. You guys know what that means? You just squeeze it, and the oil comes out of it, okay? So that's the major component of nutmeg oil. Anyways, I found out all kinds of fun things about nutmeg when I was doing my research for the lecture. I spent a lot of time on this, by the way, guys. Apparently, people use nutmeg as a drug, like induce kind of psychedelics, yeah. But then it turns out it can kill you, so I'm like,
What does it do?
I don't know. I didn't suddenly go, yeah, I have to go try that and do it. Apparently, yeah, it's illegal to like import into some Middle Eastern countries because it can be abused, and yeah, it's
Well,
weird.
like if you eat enough poppy seeds, you can test positive for opiates.
That's what they say. I don't know. So anyways, so there's palm, right? Palm trees, palm nuts. And then there's animal fat. That's where we get most of our stearic acid from. The stearic acid is also found in plants. Okay, you find it in all kinds of places. These are all different versions of saturated. And you notice they basically all look the same. They just get longer. So simple enough. You don't need to remember the sources or anything like that, but just understand that these are all fatty acids. Stearic acid, like I said, is pretty common, so it might be good to know that. Now, here's a interesting factoid. Let's see, palmitoleic. Okay, so let's go back up one, right? What is 16-0? Palmitic acid, right? Comes from palm, right? Palmitoleic in butter, right? 16-1. But it's still 16, right? So it's the same number of carbons, but the one means it has a double bond in it, okay? It turns out what oleic comes from, it comes from this word, I'll write it for you. Oops, I'll write it for you in pen. Olefin. Olefin just means oily, okay? So it turns out these are oilier. Stearate is hard. It's like brittle. I mean, if you let it warm up in your fingers, it feels oily. But these ones, like butter, like nobody has to tell you. I don't think I have to tell you what butter feels like, right? It's oily right from the start. So this word oleic, and oleic here, this just stands for it's an oily substance, okay? Stearates are harder substances. So oleic acid is the unsaturated version of which one? At 18-1, it's the unsaturated version of stearic acid, yeah. Okay, so real quickly, this is carbon 1. This is 9, right? If you count. You can count it here, too. 1, 2, 3, 4, 5, 6, 7, 8, 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. They both have their double bonds starting on the 9th carbon, okay? Now that gets to be old. When the double bond position is way back here, it gets to be old to count all the way back here. So we have a different nomenclature when the double bonds are really far to the back. I'll explain that to you in a second, okay? So the other thing to notice about the double bonds, these are known as monounsaturated fatty acids, is their configuration. Is that cis or trans? So, how do you know? They're on the same side. Like if you were to draw a line through here, like that, both the carbons, or both the hydrogens, either way you want to look at it, come off on the same side. So it turns out in most naturally occurring fatty acids, we have cis double bonds, okay? One of the things you'll learn about is that the other configuration is what you get if you heat naturally occurring fatty acids. You heat them to high temperatures, they'll sometimes go into the trans configuration. Where have you heard that? What? Trans fatty acids, right? TFAs, and are those good for you or bad for you? Yeah, those are bad for you because they behave a lot like saturated fatty acids, because that part of the double bond straightens the molecule out. It turns out if you were to try to look at this molecule and see what it looks like, and I'll show you that in a second, it actually has a kink where the double bond is. And the more double bonds it has, the more kinks, right? So if the molecule's kinky, it doesn't stack well. I know that sounds counterintuitive, but if the molecule has a lot of kinks in it, he's covering his face. Like, hold on. If the molecule has a lot of kinks in it, it doesn't stack together well, and as a result, okay, doesn't form solids very easily. So the more double bonds you have in it, the easier it is for it to melt. Okay? Talk about that more again later. But. All right, here's some polyunsaturated. Now there's linolenic and linoleic, that's the top one. And I remember this, there's a stupid way I remember this. There's linolenic, and there's, linoleic and linolenic. These are related to what? Which saturated fatty acid? Stearic, yeah, they're related to stearic acid, but linolenic has, linolenic, that's the middle one, has 3 double bonds, and linoleic only has 2. And the stupid way I remember it is when I say the word, the number of syllables that's, let's see, linolenic. Well, it has 4 syllables, but it has more syllables in it, so it's got more double bonds. I know that's a stupid way to remember it, but that's just the way it is. So, yeah, this has 2 double bonds. Linoleic has 2 double bonds. Linolenic has 3 double bonds. And now there's another pattern that emerges in unsaturated fatty acids. Okay, let's count again. 1, 2, 3, 4, 5, 6, 7, 8, 9. So the double bonds are all starting on carbon number 9 still, right? You'll notice that there's a carbon in between the double bonds. I could have had another double bond here. Turns out that's actually a better place for it. But every double bond is separated by a carbon with single bonds on it, okay? So the same thing's true here, right? There's carbons with single bonds on it. So these are all spaced out. None of them are next to each other. And that's just a pattern that we see in naturally occurring fatty acids. Arachidonic acid is one of the most important ones for pain, inflammation, and swelling because it leads to the production of things that we call prostaglandins. Okay, and so we'll get to that in a little bit. But it is a C20, okay, and it has 4 double bonds in it. This one starts a little bit earlier, 1, 2, 3, 4, and on the 5th carbon. But it's starting on an odd number, just like the other one starts on a 9. Okay. All right. Just patterns, things to recognize.
With like butter, like I can't believe it's not butter. How would that look in comparison to the actual molecule?
Like butter? Okay, butter is flavor with fat. So it's just the fats are different. So I'm not sure how they're doing it in these I can't believe it's not butter things. But they typically what they do is they take the saturated fatty acid chains. Okay, so butter is made up of triacylglycerides, has a lot of triacylglycerides in it. And what they do is they substitute some of the saturated chains with more unsaturated chains. So they make something that's technically worse for you, better for you, I guess. But I don't know that there's any real studies that show that. So my philosophy is I like butter.
Yeah.
Right. I will be the test for you when I have my heart attack. You'll be like, well, shouldn't eat like that. Yes. Sorry, just realistic, you know. I live in the Central Valley. Notice what happens, sorry. Notice what happens to the melting points in all these. Let me go back up for a second. Lauric acid, these are the saturated ones. Look at the melting points now. They're all the same saturated type of fatty acid. They only differ in chain length. And you notice the melting point goes higher and higher and higher as the mass of the molecule gets bigger, right? We'll just take stearic acid as a standard. It's 69 degrees Celsius. Your body temperature is 37, right? Degrees Celsius. So this is melts at a much higher temperature than body temperature. And that's why when you in one of my labs, we make stearic acid, and it's not this lab, not this class, but a different class. It's when the students do it right, you know they have it right because they say, is this okay? And it's a fatty acid, but it's like a rock. It's like a hard crystalline substance. Okay, so notice what happens. Oleic acid versus melting point's 14, right? Versus stearic acid, which is 69. It's a big difference in the melting point. 14 is well below your body temperature. This is how I judge fats that I eat, by the way. Is it a liquid in my body or is it a solid in my body? And I just try to avoid the ones that are going to be solids in my body.
But that's weird that butter, because butter's animal fat.
Yeah, but you have to remember it. We talk about.
And I thought butter was saturated.
Yeah, we talk about. We talk in two general sweeping terms. We say animal fat bad for you, saturated. Plants good for you, unsaturated. It's just not that clear cut. There are a lot of unsaturated fats in animals, and there's a lot of saturated fats in plants. You just have to be careful what you're eating. Plant fats tend to be better for you. Some of the shorter, I think what they're finding now is some of these shorter chain fatty acids that we're finding like in palm and coconut actually help to lower the bad cholesterol levels in your blood and increase the high density, the HDLs, the good cholesterols in your blood, lipoproteins in your blood. So it's a lot more complicated than plant or animal. I think it's a good general safe assumption to think that plant fats are generally better for you and animal fats are generally worse for you, but it's not like one or the other. Okay? Yeah, and don't fry all your food. That's the other thing. Of course, then I like barbecuing, and then barbecuing is bad because you introduce all these, they call it polyaromatic hydrocarbons, PAHs. They're not good for you. Okay, anyways, oleic acid, and now we're at linolenic acid and linoleic acid, and those are -5 and -11. And you notice the number of double bonds that you have in the molecule drastically decreases its melting point. These would definitely be more liquidy in your diet, right? Liquids as you eat them and in your body. Okay. Any questions so far? So just pointing out properties, things you should be able to recognize. I would say, I say this in all my classes, arachidonic acid is probably one you should memorize. I can't tell you why, but it always comes up. Arachidonic acid. And I won't get to that one until next time we get together. But it's one really that you're going to come up in physiology, when you take physiology, if you haven't taken it already, or if you have to take anything that involves studying analgesics like aspirin and ibuprofen and acetaminophen, and all, they all act by inhibiting enzyme processes on arachidonic acid, so it just doesn't hurt to know it. Okay, so what's the deal with the double bonds? Well, naturally occurring double bonds are in the cis configuration. Which one is this? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 16, 17, 18. Oh, so this is linoleic. Yes, it's linoleic acid. C18-1, 18 and 1. And that's what I'm trying to remember. Oh, sorry, it's oleic acid. It's 18 and 1. So it's 18 and 1. So that's what that one. That's actually what the shape looks like in solution. Okay, so if you think about a cis fatty acid trying to form a solid, right, that's that whole idea of the potato chip. Now it has this extra kink in it. It's much harder for it to stack. On the other hand, saturated fats and trans fatty acid, has a name, ellatic acid, has a trans double bond. This can occur naturally, but most often this kind of thing happens when you heat fatty acids up, and the double bond converts from cis configuration to trans. That's why frying foods, high temperatures, that's what's bad about it. It converts good fatty acids into bad ones often. Okay, so you get these trans fatty acids. This is much nicer. So think about, oh, I have to move. It's moving season. I don't have to. I'm just pretending. I have to move. It's moving season. I've got to pack all the stuff in my house. All the stuff in your house looks like this, or all the stuff in your house looks like this. Which is easier to pack? This one, right? And that's why it forms a solid much easier at a much lower, or forms a solid much easier, and it takes a lot of energy to melt it. So you have to get to very high temperatures to melt those. Sorry. Okay, so humans can synthesize fatty acids from carbohydrates or other fatty acids, okay? But we can't make enough of polyunsaturated fatty acids, such as linolenic acid. And so we actually get most of those through diet. And anything that you have to get through your diet are known as essential whatever it is. So essential fatty acids are the polyunsaturated fatty acids. And your body can make them, you just can't make enough of it. Okay? Although I always wonder, maybe we just think we need too much fat, so we just eat a lot. I don't know. I'm sure there's some truth to that. I don't know the research behind that statement, but it seems awfully fishy. I got to eat something that's fatty for me. All right. So just to give you another impression, saturated fatty acids look like this. They stack like the Pringles, right? That gives them very high melting point. Strong, we call it dispersion. Remember, the type of interaction that happens between nonpolar molecules is primarily dispersion forces, okay? And then if you look at unsaturated fatty acids with the cis double bond configuration, it's much harder to make these guys stack together. Okay? And so it's more like the stack of potato chips, although potato chips have a lot of saturated fatty acids in them. Anyways, lower melting points. Yeah, they should have picked something. Yeah, right? This was like bad, but I thought, well, they have the picture, so I'm going to use it.
I have a question. Okay, so you know how you're saying like the stuff that you eat, whatever, if it creates a solid body, is that why like you're saying about the high temperature, like when people have to work out, they have to do it for like an excessive amount to like burn that part off or something?
No, that's not.
Okay.
Yeah, it's just that my general philosophy is fats and oils that have a high melting points. Those are the ones that are generally bad for you. Those are generally the saturated fatty acids.
Yeah.
Okay, but there is actually something that I'm surprised that hasn't been said yet in I don't know if if the distinction's clear in your book, the difference between a fat and an oil. Because we all kind of think of it as the same thing. There's a difference between a fat and an oil. So, and I can get the definition out of you by asking you, like, what does an oil look like?
It's liquid at room temperature.
It's liquid, and what does a fat look like?
It's solid.
Yeah, that's the difference between a fat and an oil. If it's a fat at room temperature, 25 degrees Celsius, it's a solid. If it's a liquid, right, then it's an oil at 25. Okay, so let's just quickly do a review of what we've learned. Why is this an acid? I like this is another one of those weird trans fatty acids. Why is this an acid? That has this carboxylic acid group on it, yeah? So it has that. So it's an acid because of that. How many total carbons? Let's count them up. I'll let you count them up. How many? Is it 18? I didn't count yet. Yeah, 18. Good. Is this saturated, monounsaturated, or polyunsaturated? Mono. Because in the hydrocarbon chain, it has the one double bond right there. Wow, the highlighted was way too dark. Let's do that. And is it likely to be a solid or a liquid at room temperature? Solid. If it was not trans, it would likely be a liquid. But because it's trans and it can stack nicely, it's most likely going to be a solid at room temperature. Okay.
The trans part is which part?
Yeah, it's this double bond with the groups going opposite ways across the double bond. Yeah. Soluble in water or not soluble in water? Not, right? None of them are soluble unless they're the salt form where the H has been removed over here and you have a minus sign and some metal ion with it, okay? All right, let's get into prostaglandins. Prostaglandins come from arachidonic acid. I just thought it would be fun to draw arachidonic acid. It's not quite in the right configuration because it gets really gnarly to draw it. These are all cis double bonds and I chickened out. This one I made trans. Has 20 carbons in it, 4 double bonds. And the double bonds start on the 5th carbon. And it follows that, oh, that's a great color. Let's fix that again. Oh, I want it purple. I like that purple color. All right, it starts on the 5th carbon. It has that same pattern that we've recognized before. That is, it goes and it has a non-double bonded carbon in between each one. And this structure is wrong because this should be going this way. Okay? So that part of the molecule I need to fix. Needs to go that way and then that way. But they should all be cis in their configuration. A lot of times you'll see arachidonic acid drawn like this in biology textbooks as a hairpin like this. That's because that's the shape it gets because it has all those double bonds. It actually likes to live inside. It's one of the constituents of the cell wall. So in the lipid bilayer, and we'll talk about lipid bilayers too, it's part of this section. In the lipid bilayer, arachidonic acid sits inside the lipid bilayer, and it's one of the constituents of the cell wall. When you get injured or a cell wall is ruptured, then the arachidonic acid gets released and triggers the production of what are known as prostaglandins. Okay? So it's produced in most cells. It's very hormone-like in the sense that it does not exhibit its action within its own cell, but when it gets because the cell that it's in usually is been destroyed, the cell, it leaves the cell and has an effect away from the site of origin. Okay, so you know what like generally like hormones, they act at a distance far from their site of production. Yeah, well that's what it is anyways. A hormone is like, we'll talk about this too. You have on your kidneys you have adrenal glands, right? And then it's coming up to Halloween, so you can imagine somebody does something scary to you, and what happens to your adrenaline? Goes out the roof, right? And what happens? Your heart increases. Your blood vessels dilate so you can get more blood. I think it dilates. Yeah?
You want
more blood and your respiration increases and all that stuff. Well, that comes from a little gland on top of your kidneys. Right? So all those actions are happening away from the gland. That's how hormones behave. My God, I had that class years ago. All right, so prostaglandins differ by the substituents attached to a 5-membered ring that forms from arachidonic acid. Really quickly, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So that's the arachidonic acid. It has still some of the hairpin shape of the arachidonic acid, but it's picked up some oxygens. This is PG and E. Sorry, prostaglandin E and prostaglandin F. And if I remember right, the E stands for it's the prostaglandin that would dissolve in ether. And the F, I can't remember what it stands for, but anyway, it was a long time ago. Right. You just need to know prostaglandin E and prostaglandin F. They have slightly different solubilities. F may have been soluble in alcohol, like ethyl alcohol. You'll notice that the real difference in these two structures is what? One has a ketone and one has an alcohol. One's a little more water soluble and one's a little less water soluble. Okay. Yeah. And this little number that's down here is the number of double bonds that's in the structure. Yeah, I don't know that you have to know all this nomenclature detail, but the E and the F simply stand for what it can dissolve in. And that's back from the days of when they discovered prostaglandins and were isolating them originally. And the E is ether because in Swedish, ether starts with E, and F is something else in Swedish, and I don't remember what it is. But anyways. All right. So here's what happens. Arachidonic acid is part of your cell wall or cell membrane. The cell membrane ruptures and it releases the arachidonic acid. And then what happens is there's this enzyme called cyclooxygenase. This is not in your book, but it's oxygenase. I am writing. There we go. Cyclooxygenase, that's the COX. There's a whole bunch of these, 1 through 5 now, I think. And they're all distributed in different parts of your body. But the basic premise is when you smash a finger, you produce the arachidonic acid, the cyclooxygenase converts it into these guys. And then what these molecules do, and it's not important that you know the specific structures, is they do things like promote inflammation and swelling, because why would you want inflammation? Because they increase the blood flow. So you get damage to an area, you get an increase in blood flow, elevate temperature to fight off infections, and all kinds of things like that. Okay. So that's the general function of these. Yeah, I've got this picture of a guy hitting his thumb. I've never done that.
Is that like almost the same thing, kind of like histamine?
Yeah, it's. I'm not that familiar with his. I'll review that eventually.
Okay, that's for like allergic reactions?
Yeah.
Yeah, that's what it was.
I don't think it's totally unrelated, though. I'd have to go look it up, though. Okay, so cyclooxygenase is this enzyme here. Wait. And what the analgesics do, like ibuprofen and acetaminophen and Tylenol do, is they mimic the shape of arachidonic acid, and they sneak in there and they keep the arachidonic acid from getting in there. So they're known as competitive inhibitors because it's like, if you can put cyclooxygenase in that enzyme, then you can block the production of prostaglandins and then reduce inflammation and pain and all that kind of stuff. Because the distribution is different and because these drugs don't all act the same on each enzyme, you get different effects for like Tylenol and for ibuprofen. Right? And so some people swear that they'll take ibuprofen for a headache, where some people say, well, it doesn't work for me, I have to take Tylenol. It has to do with the kind of headache that they have, whether it's like a muscle stress headache or inflammation, swollen joint kind of thing. Okay. Aspirin is an interesting one. Aspirin is known as a non-competitive inhibitor and actually bonds to the cyclooxygenase, and you only need one to get in there and it blocks it. Okay. I think I also mentioned earlier that cyclooxygenase in this whole process also is responsible for the mucus lining in your stomach, and so when you take a lot of these non-steroidal anti-inflammatories, the NSAIDs is what we call them, these guys up here, they were in the news a few years ago because people were having heart attacks or something. Sorry, I should have kept more track of those things. But what was I going to say?
Stomach lining.
Oh yeah, stomach lining. Yeah, you stop producing the mucus when you take these things, and so basically the stomach acid that you produce just starts digesting your stomach lining. Yeah, yeah, sounds bad, huh? It generally is.
That's only if you take like way too much, right? You can take
Or you could take just a regular amount for a long period of time. It'll do the same.
empty stomach.
Empty stomach, yeah. Because when you eat, you produce mucus lining in your stomach. Then that's why they say you should take ibuprofen, you shouldn't take it on an empty stomach. Yeah. Which I do all the time and I pay for it. I'm like, oh, I'm too much hurry. But when they say eat, okay, how much do you need to eat?
Like a toast.
Like a few pieces of cracker. I mean,
Oh.
yeah, you don't need to go, man, I need a whole turkey sandwich and roast beef. And then I'll be okay. Yeah. Yeah, anyways.
Wouldn't it take longer to work too?
Yeah, I don't know.
Again, if you had a lot of.
Yeah, but it's just when you get all that fat into your system and it sort of, those receptors all fire in your brain, maybe you don't need it anymore. All right, so last thing to cover, we're going to talk about omega-6 and omega-3 fatty acids. And this is one of those things I was really proud of myself for figuring out in getting ready for lecture. I think I mentioned this the other day. So, so this is linoleic acid and arachidonic acid. We often refer to these as omega-6 fatty acids. So if you think about the Greek alphabet, what's the first letter? And the last letter? Omega. So alpha and omega, those are the first and the last, okay? When we say omega-6 fatty acid, we do that so we don't have to count from the other end. Omega, starting at the last carbon, minus 6. Okay? So you go 1, 2, 3, 4, 5, 6. That's the position of the first double bond, so it's an omega-6 fatty acid. There's also omega-3 fatty acids. Right? Unsaturated fats recognized as more beneficial to health than saturated fats. We find them in vegetable and fish. And examples are linoleic and arachidonic acids in vegetables. But yeah, you can eat fish and get a lot of omega-6 fatty acids. That's what they're referring to. But really, it's just like linoleic acid. You can get it from a plant too, I guess. Percentages maybe aren't as high.
Now, those are the ones where they said, like, if you cook them too high heat,
If you cook and then there'll be trans fatty acids, then you're kind of
it's not good for you.
not good for you.
Do you know the temperature?
So actually what my mom taught me to do is to cook salmon in the microwave. So you take salmon, like a piece of salmon. I went to Save Mart the other day, and there was salmon, and it was $1 a pound.
What?
It was like today's special, and it looked like it had been frozen and then neglected for a while, but I thought for $1 a pound, I'm going to try it, and it was good. It was imported from Chile or whatever, and I know they had some problems over there. But we got it. You just take it and you stick it. I know, horrible, right? You stick it in the microwave. You put butter and garlic on it, and it's like 3 minutes or 5 minutes. I think we do 3 minutes, and it's a piece of salmon about that big, and you cook it for like 3 minutes in the microwave. It's done.
It cooks in 3 minutes?
Yeah, something like 3 minutes.
Right, so
You can.
how hot would that change?
Well, microwave boils water, so it's like almost like poaching it. You can burn it, of course, right? You can burn it, but I don't think it gets that hot, and it's done in just a few minutes. So part of the converting it over is temperature and its time. So the way my mom taught me to do it is you just put a piece of salmon in a like one of those glass lidded containers. Put butter, garlic, throw it in the microwave. 3 minutes, pull it out. It's done. And yeah, it was kind of a weird thing to eat $1 salmon, but my whole family ate it. None of us are dead today. So here's a couple of interesting fatty acids, or I think they're interesting anyways. These are very highly unsaturated fatty acids that apparently come from fish, according to your book. But these are omega-3 fatty acids. This is 1, 2, 3. 1, 2, 3, 4, 5. Right, double bonds. So that's why this is an ICOS. ICO means 20. Okay. pentaneoic, so that means 5 double bonds, acid. And then, oops, this DO, cos means, again, 20. DO means 2, so 20 and 2, this is 22, okay? Hexaneoic acid means it has, that's this one actually. No, it's this one. 1, 2, 3, 4, 5, 6 double bonds. Yeah, so fish have cold water fish are a good source apparently of these omega-3 fatty acids, which is presumably better for you. Yeah, that's what they're trying to get. Omega-3.
Well, they just say like omega-6 is really easy to get. It's the omega-3s that are harder.
Yeah. But at $1 salmon, I'm just saying. I just figured it was worth a shot. It's one meal, whatever. Okay, so I think we'll stop there. We'll talk a little bit about heart disease when we get back.